Hello, welcome to Fun with Flags. Wait, welcome to Fun with Genetic Markers to Estimate Population Relatedness and Migration. That's kind of a mouthful, isn't it? Let's just see if we can unpack it. I'm going to be reading for part of this because I am not that good. A genetic marker is a gene or DNA sequence at a known location on a chromosome that can be used to identify certain characteristics. And in some cases, this marker can be for a characteristic that is also observable in the organism. For example, eye color, blood type. We can look at the genetic marker for that trait and say, there it is, in the DNA, blue eyes. We can also just look at the organism and say, yes, that person has blue eyes. But there are also genetic markers for things that do not show up when looking at the organism itself, because the trait that the marker is marking is not being expressed. Either it's not. A really good example of this, actually, is breast cancer in women. We know that breast cancer is largely hereditary. Women that get breast cancer, it has always existed in their families. But women who have breast cancer in their families do not always get breast cancer. So there's no way to really tell. It's a crapshoot. But with genetic testing, we can look for the marker for breast cancer. And women who have it in their family, we can look and see. And even if the woman is not showing symptoms and does not have cancer, if she has that marker for cancer and has it in her family background, she is pretty much going to develop breast cancer. So the marker allows us to see a trait that has not yet expressed itself. So having a genetic marker for a disease like breast cancer allows people to make better choices about their health. Now, in terms of, as we're talking about here, right, population relatedness and migration, one of the things that we can do with genetics is to find out the degrees of relatedness from species to species and of individuals within species. So in terms of species to species, for example, that helps us decide where to classify an organism in the typical Linnaean Latin classification system I have. Just by every couple of years, suddenly an animal's Latin name will change because DNA has allowed us to see that that animal does not belong in the category we put it in. And a lot of the time, this is very much counter to what it appears to be. For example, if you look at a cheetah, if anybody's ever worked with cheetahs, at some point someone always says, what's the difference between a cheetah and a leopard? And it always makes you want to slap them. But you look at a cheetah, if you don't know, you look at a cheetah, and it's a large spotted cat. And you think, okay, it's got to be closely related to a leopard, also a large spotted cat. They live in the same area, live in Africa. But then it turns out, genetically speaking, that cheetahs are not particularly closely related to leopards. So that's sort of how you use genetic markers to understand relationships between species. Things really, really get interesting, though, is when you start looking at genetic markers to figure out how closely related individuals are to each other. This is really important, both in captive breeding and in management of wild populations. And the reason is that in a captive population, the way that we keep track of who's who is every zookeeper has access to the stud book for the species that they're working with. And the stud book contains all the genetic information, the inherited information we have about every animal in captivity. So we know who's related to who, whose sisters, whose fathers, whose mothers and daughters, so that we can prevent accidental inbreeding. And this works really well. It's worked really well for a long time to really keep track and make sure that we have enough genetic diversity in a captive population. However, there are problems. One of the big problems is that if we have all this information, we're golden. But if we have an animal that is not in the stud book, that we do not have a history on, we don't have any way of knowing what or who they might be related to. And therefore, that means that especially in a species where there's limited genetic diversity to begin with, we can't bring in new genes because we're not sure if they are new. We bring in another animal that we don't know its pedigree, and made it with an animal that we have. It may turn out that they were brother and sister, and we just didn't know, or more likely that they were cousins or whatever, and we didn't know. So the other problem becomes with if, for example, we have a very small population of animals in the wild that we bring into captivity for breeding purposes, like say we do with the Sumatran rhinos or the condors, that you bring the population into captivity to breed, but then you don't have any way you can really tell who in that population is related to who else other than sort of looking at them. But, yay, we have genetic markers. The genetic marker, you can tell what individual is related to what other individual, even if you do not know their history. If you don't know who their parents are, you don't know who their siblings are, doesn't matter. Genetics don't lie. You can figure out the degree of relatedness, and we talked about degree of relatedness, between two individual animals, and therefore can figure out which animals should be allowed to breed and which shouldn't in captivity. Now, in a wild population, it's even more exciting, right? Because you pretty much never know what's going on in a wild population. I mean, you could observe them and sort of see who's mating with who and that kind of stuff. But in general, when you're looking at a wild population, it's very hard to tell what the family relationships are and how to track who's doing what. But with DNA sampling, you can, and it's really easy. Quiet, dog. It's really easy because you don't even need to dart the animal and take a blood sample or anything like that. You can get DNA from a tuft of fur or a pile of poo or whatever you want to get DNA from. And this allows us to not only know who's related to who, but we can figure out where they have gone. So if you have a population of animals over here, and then you have a population of animals over here, and you think, ah, yes, these are two distinct populations, but you find genetic markers in this population over here from this population, then you know, even if you didn't see it, that genes are migrating from one population to the other, which means specifically, an animal or animals are moving between populations for mating purposes. That's giving you a lot more diversity than if you had two completely separate populations that didn't cross. If you do have populations that don't cross, there's no matching genetic markers, then you can think about, well, do we need to build, say, a wildlife corridor? We have that going on here in Southern California right now. There's a population of mountain lions that's completely hemmed in by freeways, and they can't get out in order to mate with other populations of mountain lions. So they're becoming inbred. But we can tell one of them made an escape. P22, he's very well known, moved from the Santa Monica Mountains into Griffith Park. And we know it's him because he's radio called, but we can also track his genetic information. So if we can get a girl to follow him to get into Griffith Park, then we'll have beginnings of a second population of mountain lions, and we'll be able to track who is coming and going across a wildlife bridge, if we ever build one, by following genetic markers. There's a lot of really, really exciting stuff going on. And it gets sort of more exciting all the time. So I just realized we're also supposed to be doing drawbacks of the stop of genetic markers. So I wanted to go over a couple of those. One of the biggest ones, of course, is expense. It's equipment is expensive. You need to have someone with expertise. Things are a little more confused today than they were earlier. So there's some drawbacks in terms of using it in the field, just because of it's more complicated than just doing, say, straight out field observations. It requires money and time that is sometimes at a premium in research. But I think, hi, this is Dottie. We just got Dottie. She doesn't have any manners. I think that problem will resolve itself in time, because that's the way technology is. It starts off sort of very esoteric, expensive, and difficult, and then over time it becomes standardized and it sort of comes down in price and becomes sort of something that people are more familiar with and are more trained to use just coming out of the gate. So that's an issue, but I think, again, it solves itself. The other issue that is a little bit bigger is that genetic markers can tell us a lot about who's related to who, and therefore from that, other dog, he has manners, from that we can then tell, we can track the migration of animals and their movement across the landscape. But one thing that it's not good at is taking that information of who's related to who and scaling it up in order to get sort of a generalized picture in a very large population of overall degree of relatedness. That's a goal. We would like to do that. But right now we don't actually have the theoretical framework in place to be able to scale it up properly. In order to do that, you need a theoretical model, a computer model that you can use to scale up the data that you have. And there's a bunch of different theoretical models being used right now, but they have varying degrees of accuracy. There's one study, I have the citation at the end here, that discovered that there is, between the theoretical model and the actual genetic testing, there was a difference of 3 orders of magnitude. So a lot of these models are not very accurate, and so we don't have a lot of ways to scale up this information and make it more useful to us on a larger scheme of things. But again, this is something that will resolve itself in time. We're still working on figuring out the proper theoretical models. One of the problems we have is that most of them, you have to assume something to begin with, and a lot of them assume a constant degree of relatedness or lack of relatedness in a population. That's actually not true, because there'll be clusters of relatedness across the large population. So a lot of it is just, we just need to figure out the appropriate framework or modeling we can use to be able to scale up. We just haven't got it yet. And there's so many studies out there, I became very overwhelmed. I was going to try and talk about a bunch of them, but it's just too much. So we're still working on that. It's a really exciting time to be studying biology, and actually it's an exciting time to just be living in the world, because we have all these tools in our toolbox now we didn't used to have. We have all these ways of figuring out how we live in the world and how we help other creatures live in the world with us, and how we go about our lives in a way that causes as little damage as we possibly can. It's a really great time, to be doing what we're doing, and getting chewed up by a bull terrier. Yeah, I wanted a bully breed. I don't know what got into me. So anyway, that's it. One last thing I wanted to share with you. This flag that I held up at the very beginning of all this, I just wanted to let you know, this is the International Naval Flag. That means I am on fire and carrying dangerous cargo. Please beware.